Oh, and I just got this meeting is being recorded, so that's good. It's a pleasure for me as one of the protein folding disease seminar speaker hosts to have invited Nick Testakis to give us a talk about his interests that relate to autophagy. Now, my own link to the protein folding diseases program is through the ER Hub, and my real dream was that we're going to learn everything about phagy. And Nick immediately said, you know, I'm not talking about that. So that's fine. I mean, the issues that we each confront have great similarity in the macroautophagy field. And so I do believe that the kinds of things that Nick has been pursuing about mitochondrial autophagy, mechanisms of autophagy are going to have relevance kind of across the board. So I do believe that's going to be interesting for many of us. Nick's I know that I met Nick at exactly one time previously in my life. It's, you know, like almost before the rest of the audience was born. But Nick did training, got PhD training in Texas, at University of Texas, and did postdoctoral work with Mike Roth at Southwestern. And that is actually the period of time in his previous life where we had once met. He has a long and illustrious career and has transitioned from lipids into this whole area of organellar autophagy. The Protein Folding Diseases program has obvious interest in these kinds of questions as it pertains to cell viability as well as organelle turnover. So this is exactly the reason why I was hoping Nick could give us a talk today. What I see is that there are 47 people. I don't know who's out there, but what Nick has said is that we should try not to interrupt during the talk, but type our questions into the chat box. And then at the end, we will save time to run through those questions. So as things pop up in your mind, you know, either make a note or type them into the chat box, and we'll cover the whole thing at the end. So with that in mind, Nick, I'll ask you if you can share your screen and you can begin.
Absolutely. Can you see okay?
Yeah.
All right, so let me just make a presentation. So thank you very much, Peter, for this nice introduction. I hope I will not lose any of the audience if I start by saying thank you, Michigan, and not just for the invitation to me, but for other reasons. So I want to talk to you today about the work in my lab. I will finish with the work on mitophagy, but actually I will start by describing the work that we have done looking at non-selective autophagy. And the work has been done by a very talented group of students and postdoc and research scientists shown here. And I also want to highlight three very important collaborators: Lucy Collinson at the Crick Institute, Roger Williams at the LMB, and Eva-Liisa Eskelinen in Finland for a lot of the work. I work at the Bebraham Institute, which is an institute outside of Cambridge. It has connections to the university, and it's a very old institute, but not as old as this picture here. So this is actually a stately home that was then transferred into the government, and it became a research institute. So my thinking about autophagy is as follows. It is a cellular response to either nutrient limitation or stress. And nutrient limitation can be in the form of absence of growth factors, amino acids, or even developmental stage. And stress could be the presence of damaged proteins or organelles, and or the presence of pathogens, a virus or a bacterium. And depending on this stimulus, autophagy either generates nutrients if the stimulus, the limitation of nutrients, or it mediates a quality control pathway, and both of these lead to survival. Because on the other side, and responding to the same kind of signals, is apoptosis and cell death. So autophagy is a very important in my opinion, survival pathway. And I think you probably know the basic parameters, but I will just go through them very quickly. So the idea is that the cell makes a novel double membrane structure in the cytosol, which encloses material. And so this is the beginning of this process where the double membrane starts to form. It's also called the phagophore. In the process of this, cytosolic materia is enclosed, the membrane expands and closes, and then, so this is where the membrane closes, and this is the autophagosome, a double membrane structure. And then from this point on, the autophagosome starts to interact with the pre-existing cellular machinery, as far as we know, in order to mature and eventually to start fusing with the lysosomes. And that leads to the degradation of whatever was inside the double membrane structure, either because the cell wants to get rid of it or because it wants to recycle components, for example, amino acids. And my lab has been interested almost exclusively at the very early steps of this pathway. So how this early membrane is formed and how it starts to expand. So my discussion will be mostly focused on these very early steps. Now, because of the focus of your consortium, I put this slide in that shows various genes that have been associated with neurodegenerative diseases and also are implicated in autophagy. And they are implicated in basically all of the steps that I just showed you, from the very beginning, the formation of the early membrane to the formation of the full autophagosome, and then the fusion steps with the lysosomes. And this is the genes that have been implicated. One characteristic is that most of them are not the canonical essential autophagy genes, but they are actually peripheral regulators of autophagy. But there are some, for example, here ATG5. Another group of genes is the ones that are involved in selecting cargo for selective autophagy. And in this category, here we have P62, optineurin, TBK1, and I will talk about these regulators a little bit later. So autophagy, because it can clear aggregates, I think it is important for neurodegeneration as well. So the nuts and bolts of the pathway are summarized here. And basically, the pathway starts from the inactivation of a protein kinase called mTOR. The complex is called mTORC1. That leads to the activation of a kinase complex called ULK complex that has 4 proteins. This complex, and in the presence of ATG9 vesicles, the complex and the vesicles translocate to a region of the endoplasmic reticulum or other membranes, where they activate another kinase complex, but this one makes a lipid, not a protein. So this is the VPS34 complex that makes PI3P. And the PI3P starts to accumulate in a novel compartment, which we have called omegasome. And within this compartment, we see the formation of autophagosomes. And the process of autophagosome formation involves a very complicated set of reactions that end up with a covalent modification of a protein called LC3 with phosphatidylethanolamin, and that basically decorates the membrane of the autophagosome as it forms, and eventually the autophagosome is free to go and fuse with the lysosome. So this is the pathway. I want to pick a few parts of this diagram in order to expand a little bit in my talk. So I will tell you a little bit about how we think the ULK complex goes to the membrane to start this process. We think that's a very important early step. I want to describe to you some older work that sort of defined what the omegasomes are, and in the process tell you a couple of things about the VPS34 kinase complex. And also I want to touch upon a second very important function of autophagy, which is selective autophagy, and that is the formation of the autophagosome in response to the presence of a specific cargo, for example, in this case, a damaged mitochondrion. And the work that I will tell you about has all been published in the last few years, so you can see the publications here. So let's start from the step where the ULK complex from the cytosol goes to find the membrane in order to start the process. And the problem with trying to dissect this step is that we don't know what this early structure looks like. Autophagy is very nice because you can go into a cell that is undergoing autophagy and you can basically find all of the structures that I just showed you, and sometimes very close to each other. So you can find the early membrane as it expands, you can find the full autophagosome. But the problem is that we don't know what the ULK complex looked like at the very beginning. So in order to address this problem, we went into super resolution microscopy. So we used STORM in order to find what the early structures of the ULK complex look like. And we use endogenous antibodies against ATG13. And this is what we saw. So we saw this kind of structures forming in the cell upon induction of autophagy. And they looked like they were in some kind of a progression, although these were fixed cells. So we were not sure about this. But it was this kind of multi-subunit structures that were forming. In fed cells, we saw much smaller structures. If we inhibited the BPS34 step, so we only allowed the very early step to take place, we saw this sort of like mini assemblies. You can see those two examples here. And then when we looked at this kind of structure or this kind of structure in connection to the endoplasmic reticulum, what we saw was basically that the ULK complex was forming at extensions of the endoplasmic reticulum, or sometimes it was sort of connecting. You can see here the endoplasmic reticulum strands. So armed with this kind of result, then we went and set up some live imaging experiments because we wanted to see what actually this looks like in real time. So what we try to do is to combine live imaging and super resolution microscopy. And this is an example here. So here cells are expressing ATG13 and an ER marker, and they are in the process of being starved. And if I play this movie here, you can see the formation of an early ATG13 structure. And then a little bit later, another one forms. And because we have the time element here, we know that this is about 2 minutes old, whereas this one is about 10 to 15 seconds old. So then we fix the cells at this stage, and we find these structures. And first we look at them by SIM, which is another super resolution technique. And these two structures here, you can see them where they are in connection to the endoplasmic reticulum. So they're basically almost completing the endoplasmic reticulum strands, you can see here. And if we look at them by STORM, the more high resolution technique, you can see that the older structure is basically this collection of these little globular elements, whereas the very early structure here is just the beginning. So these are basically starting to come together. We used another technique to look at this in more detail. And again, we set up live imaging, then we fixed the cells, then we used focused ion beam microscopy, because that gives you basically the slices through the whole cell. So when we identified an ATG13 early structure, what we saw, so this is the green here, what we saw underneath this is basically a collection of tubulovesicular elements. And this is shown in this reconstruction here, where you can see the ER is in green, there is mitochondria in blue, and the ATG13 structure is inside here in red. And this is more structures of this. So you can see that the ATG13 or the ULK complex structure is forming in very close association with AR, although it's not really ER because you can distinguish the AR from it, but it is very, very tightly connected with AR. So in our opinion, the earliest structure that gives rise to an autophagosome is this tubulovesicular compartment that is related but distinct from the AR. It's demarcated by ATG9 vesicles. I didn't show you the evidence for this. And it contains the ULK complex. And we think this is how the process starts. Now, the omegasome is the structure that forms a little bit later. And as I told you before, it's enriched in PI3P. So in order to understand what we mean by the omegasome, I want to show you a sort of classical view of autophagy, which is shown here, where you can see a little membrane in the cytosol that starts to expand, and then eventually it closes and it traps inside it the cargo that was in the cytosol. But in our opinion, this is not a complete story. We think that actually associated with this very early structure is a novel membrane compartment, which we call the omegasome, which is enriched in PI3P, and it is connected to the endoplasmic reticulum. So another way to depict this is shown here, where we imagine that in a region of the AR, we start accumulating PI3P in response to the autophagy signal. This region then expands, and it's basically two separate compartments. The outer compartment is the PI3P enriched membrane, which is green here. The inner compartment which is red, is the collection of the autophagy proteins. This expands, and at some point it starts to bud inwards, eventually liberating the autophagosome, and this structure diminishes and gets reabsorbed back in the AR, and then the process starts again. And where does this come from? It comes from some live imaging experiments that we did a few years ago, where we imaged the PI3P compartment with DFCP1 in green, the LC3 protein in red, and the ER in blue. And here is the overlay. And if I play the movie first, you can see in the overlay the formation of a green structure and a red structure inside, and then the red basically comes out of the green. If I play it a little bit slower and just stop it at several places, you can see at the very beginning the formation of this PI3P enriched compartment at this piece of the AR. Still the LC3 is not there. If I continue to play, then you can see the LC3 starting to form within the same region, and they are still tracking the AR. Then at some point, this reaches the maximum diameter, and you can see the omegasome on the outside, the LC3 inside. And then if you look here, you can see that the red structure is going to shoot out of the ring. So here you can see that most of the red structure is now outside of the ring, and the ring is smaller and sitting here on this piece of ER. And so eventually the structure goes away. So we think that the omegasome then is a transient intermediate that forms before the autophagosome proper forms, but the autophagosome proper forms within this omegasome region. And if I go back and I showed you this very early step, so you see the PI3P forming at this little tubular extension of the ER. So when a collaboration with Roger Williams, we solved the crystal structure of the complex 1 and the complex 2. So these are the two VPS34 complexes. One of them is involved in autophagy. The other is involved in endocytosis. The complex 1 is involved in autophagy, the complex 2 in endocytosis. And the biggest difference between those two is the opening of the structure. So this is much bigger than the autophagic VPS34 complex. And Roger did some very nice experiments showing that the VPS34 complex that's involved in autophagy only works on vesicles of very, very small diameter. So it likes to work on highly curved membranes. And that is very consistent with the data that we had from the live imaging, where we saw that the earliest PI3P accumulation was actually on a tubular extension of the AR, which is actually a place that has a very high curvature. So that was all very consistent. So let me go a little bit out of this now and talk about the autophagy versus mitophagy. But before that, I want to spend a couple of minutes to talk about the plasticity of the autophagic pathway, because this pathway can make many different sizes of autophagosomes. So this is a typical sort of like textbook autophagosome. You see the double membrane in this clear area, and inside is the cytosol, and you can actually see a little bit of ER on the outside. So this is a sort of typical autophagosome that is engulfing basically cytosol. You can also have an autophagosome in the process of forming here that is starting to engulf a piece of mitochondrion. It already has a piece of ER inside, and there is also ER on the outside. So the sizes here are very similar. But here is another autophagosome. So this one is formed when cells are undergoing mitophagy. And you can see that a lot of little mitochondrial fragments are enclosed. And look at the size of this in comparison to these two. And we can go crazy and look at something like a Salmonella infection. So this is cells that have been infected with Salmonella. And the cell is making these gigantic autophagy structures that are enclosing actually a large number of bacteria stuck together, either in this orientation or in this orientation. So it's the same machinery, more or less, making this kind of very different structures in size, but also a little bit in morphology. So I hope that by the end of my talk, I will give you at least one possible explanation how this can happen. And again, the machinery is very similar, but in this case, we have to add an extra layer, which is basically the proteins that are responsible for recognizing the cargo that gets incorporated during selective autophagy. And this can be ubiquitinated proteins being recognized by P62, damaged mitochondria, again, ubiquitinated that are recognized by the adaptors, or even bacteria that are sometimes ubiquitinated, but not always, and again, recognized by the adaptors. And what the adapters are doing, they are basically connecting the cargo with the LC3 that's sitting on the autophagic membrane. And there is quite a lot of different adapters. And again, this pathway is very, very, very heterogeneous. You can see the kind of structures that are targeted by selective autophagy. It can be as small as 10 nanometers, which is a protein aggregate. It can be as large as 5,000 nanometers, which is a chloroplast, or an ER membrane, since you're interested in ER phagy, or something like 1 to 2,000 nanometers, which is mitochondria. So the cargo can be very different. The size of the forming structures is very different. And let's consider mitophagy as an example of this kind of selective autophagy. The other thing that we should say about mitophagy, and I think this is going to be true for other types of selective autophagy, is that there isn't just one kind. So there's basically 4 different types of mitophagy using different types of recognition of the damaged mitochondrion, different set of adapters. But in all cases, the idea is that the damage needs to be recognized somehow and then mediate basically the formation of the autophagic membrane around the damage. So this is the pink parking pathway that has been, very nicely explained by Richard Yule and other people, where you have ubiquitination of the mitochondria, and then via parking, and then receptors recognize this and they make the membrane. There is mitophagy that doesn't depend on ubiquitin, but depends on the presence of FAND or NICS proteins, and they are recognized by LC3 by the autophagic membranes. If you deprive cells of glucose, there is another type of mitophagy which doesn't use adapters as far as we know, but it uses RABs. And then a process that we have studied, which is downstream of treating the cells with a drug called ivermectin, that's basically, it uses ubiquitin, but it doesn't depend on pink parking. So it depends on other E3 ligases in order to ubiquitinate the mitochondria and then generate a membrane. So in this kind of mitochondrial autophagy, I will show you here an example. A cell is expressing the mitochondrial protein LC3 and ATG13. And if we treat the cells with ivermectin, you can see the mitochondria fragment, and a little bit later the LC3 starts to form. And every time LC3 is formed, there is an ATG13 formed beforehand. So this is a classical pathway that follows the hierarchy of the steps. There is a lot of LC3 type 2 forming during the process because this is actually a pretty robust mitophagy response. And it does degrade mitochondrial protein shown here. And it is a proper autophagic response because you can actually see during this process mitochondria being surrounded by double membrane phagophores or autophagosomes. So I want to show you a few movies that basically compare and contrast the process of non-selective autophagy and the process of mitophagy. And here is an example of a cell that expresses LC3, DFCP1, the PI3P marker and the ER. And if I can play this movie, you can see sporadically the formation of these structures that start being green because the omegasome forms first, and then the LC3 forms. And you can see this is a very sporadic, it's not synchronized at all. It forms in different parts of the cell. A very similar thing is true for mitophagy. So here the cell is expressing LC3 in red, DFCP1 in green, and mitochondrial. And you see already here the mitochondria have been fragmented. And if I play the movie, you can see mitochondria being engulfed. Every time you see a green and a red structure, there is a mitochondrion underneath. And again, you see it's a very sporadic process, just like in non-selective autophagy, but are the dynamics similar? So just to remind you again about the non-selective autophagy. So we see the formation of the omegasome, and then the LC3 comes out. And this is what we have called the extrusion step, when the autophagosome actually leaves the sort of cradle where the omegasome is. This is shown maybe a little bit better here. If you look at this, so this is just the DFCP1 and LC3, and you can see the LC3 coming out of the omegasome. This is an example of mitophagy. The mitochondria that's being targeted is this one here. And again, the cells express DFCP1, LC3 in the mitochondrion. And you can see here that the machinery attacks the mitochondrial fragment. And then you can see the omegasome going around and then the LC3 forming. And eventually you end up with an LC3 that has engulfed very precisely, actually, the mitochondrial fragment. Another example here where you can see a little bit more clearly the extrusion step. So if you focus here, you can see as the mitochondrion is being engulfed, the omegasome coming out of them. The reason why the extrusion step is important is because it basically allows us to understand a little bit that although the ER is connected to the forming structure, it's not the same structure. And actually, recent data from many labs has shown that the bridge between the ER and the forming omegasome, which then gives rise to the autophagosome, is this protein ATG2, which is a lipid transporter that basically we think transports lipids from the ER to the forming structure. The other reason why this extrusion step is important is because it allows this machinery that's forming the autophagosome not to become part of the final structure, but to be able to be recycled. Because otherwise, every time you form an autophagosome, you would basically be destroying a lot of the structure that gave rise to it. So we think that's an important step, and it explains a few things about the early steps. Now, what about the ER? So I show you another example here where we have stained cells with the ATG13 in red and the ER in green, and this is in non-selective autophagy. And if I play this, you can see the ATG13 structures forming always in very close association with the ER. This is the omegasomes in the ER, and you can see that the omegasomes, and this is TERF imaging, you can see forming very close to the strands. And in fact, if you look at the omegasome and the underlying ER, you can see that they basically have a very similar morphology. And work that comes out of Eva-Lisa Eskelinen and Tamotsu Yoshimori has shown that actually the ER and the forming autophagosome are very closely connected with each other. And in fact, the autophagosome is sandwiched between two layers of the ER. So it has this kind of very intimate relationship. So this is for non-selective autophagy, but what about mitophagy? And here we got a surprise because we didn't think actually that mitophagy would follow the same kind of rules, but in our experiments it did. So here we have ATG13 in red, we have the ER in green and the mitochondria in blue. And if you follow the red structure here, which is starting to enclose this piece of mitochondrion, I'm going to play this movie, but then stop it at several places. So you can see now the ATG13 engulfing this piece of mitochondrion, but also look at the ER underneath. So it's basically following the morphology of this engulfment step. And if I play a little bit longer and I stop it again, you can see again that the ATG13 that's engulfing this mitochondrion is sitting on this piece of the ER. You can see the rest of the ER is very different, but actually the structure here is very similar, and so on. So we can follow this all the way through, and we can see the ER actually providing the same kind of spatial clue to where the structure is forming. And in fact, another thing that's kind of interesting is that although the ER is very mobile and very dynamic, it moves around quite a lot of distance, you can see that the ATG13 always follows this, and the mitochondrion is also following it. So it's a very tightly connected structure. So the ER, in our view, provides a similar kind of clue. We looked at it not in live cells because we don't have a lot of resolution there, but again by super resolution microscopy. And again, we found a lot of examples where a piece of mitochondrion was being engulfed by an ATG13 structure. We could see other autophagy proteins, for example, WIPI2, in the same region. And also we could see that the ER, where this formation was taking place, was actually in the same region and in the same kind of shape. And even in cases where, in the bottom here, where you see the ATG13 is not fully engulfing the structures yet, but it is partially engulfing them, you can see that this still is happening within this ER region. And down here, you can see actually how tightly connected is the ER and ATG13 or the ER and WIPI2. And this is additional examples where we have looked at the selective autophagy adapters or receptors, for example, NDP52, TAX1BP, optineurin, and a kinase that's very important, which is the TBK1 kinase. And again, in all of these cases, you can see the mitochondrion that's being targeted by this machinery. And always you can see that they are is in the same region, and it's sort of like providing a special clue about where this is going to take place. And then in collaboration with Eva-Lisa Eskelinen, we did a combination of live imaging and electron tomography to actually look at this in more detail. And this is an example of a cell that's expressing the mitochondria in red and ATG13 in green. And we start by live imaging in order to find an event. So the event here is going to be coming up, this one here, I believe it's this one here. So we stop, so we freeze the cells, we fix the cells at this point, then we find them in the EM, and then Eva-Lisa and Siggy, her student, reconstruct this region by tomography. And this is shown here, and the mitochondria is in brown, the ER is in yellow, and the phagophore, the early autophagy structure, is in green. And if I play through this movie, you can see that the green phagophore surround very tightly the mitochondrion, and the ER is the vicinity of this as well. And this is a reconstruction here. If I stop it somewhere here, what I want you to notice is that there's actually 3 phagophores that surround this mitochondrion. And I will get back to this a little bit later. But also look where the ER is. It's basically sitting where there's empty area. So we think actually the ER is providing very important information that helps this structure being engulfed by the phagophores. And the question that we asked is, if we stop all of the steps except the ubiquitination step, which is the first step in this process, will the ubiquitinated mitochondria still associate with the ER? So in order to do that, we took cells that were knocked out for FIP200, and we also used an inhibitor of TBK, a pharmacological inhibitor of TBK. So we basically blocked the pathway except for the first step, the ubiquitination step. And when we look at the mitochondria that were ubiquitinated, what we saw is that they were still being surrounded by pieces of ER. So here is 3 different examples. So we concluded from this, based on a lot of other experiments that I'm not showing you, that the damaged mitochondria are first ubiquitinated, then they are encased in the ER strands, which then provide the platform for the formation of the mitophagosome. So this is the structures involved in mitochondrial autophagy. What about the ATG13 in this process? So we found very surprising dynamics in this. So let me just show you first a movie that we took every 10 seconds in cells expressing ATG13, LC3, and mitochondria. And if I play this movie, so this is the ATG13 helping the LC3 form around the mitochondrion. And what I want you to see here is that although the LC3 makes a pretty stable structure around the mitochondrion, if you look at what the ATG13 is doing, it's going all over the place. It goes from one side, it goes on the other side, it goes around. And another example here, but this time we're not looking at ATG13, we're looking at optinurin, one of the mitophagy adapters. And again, optinurin is here surrounding this mitochondrion that you can see being engulfed by LC3. And again, you can see very, very strange dynamics. And if we put together ATG13 and optinurin, then basically we see 2 structures that are targeting the same mitochondrion, but they're doing very different things with each other. They're both, you can look at this here, they're both going around, but in very different ways. So we realized that what this was telling us is that we were basically sampling the movies at a very low frequency, and we were missing basically important information, what was going on. So when we did fast movies, what we saw is that this very strange ATG13 engulfment dynamics was basically oscillations around the mitochondrial structure. So if you look at this here, you can see 1, 2, 3 times that the structure is being targeted, and then the ATG13 disappears, and then it appears again. So we decided to look at it by trying to model what's going on. And the first thing we did is we collected a lot of these examples where we could see oscillations of the ATG13 structure on the mitochondrion. So here is a movie that has 2.5. There is another movie here. There's another movie. We collected a lot of movies. We lined them up together, and we got this cumulative graph here. And then we came up with this model to explain this kind of oscillatory dynamics. And the model is that when the mitochondrial fragment is being targeted by autophagy, you start by first translocating the early machinery, so the ULK and the omegasome structures, that leads to the translocation of the LC3 that starts to form. And then we hypothesized that the ULK and the omegasomes come off, and then they translocate again at a different place where the LC3 is not. And then another piece of LC3 is stitched there, and then another translocation, another piece of LC3. And so eventually you basically cover the structure, but with pieces of LC3 that have been stitched together. And the clue for the translocation is basically where the ULK and the omegasome complex goes together. And when we make this model, we basically simulate this kind of behavior of ATG13, which is pretty close to what we observe experimentally, including the fact that as the available space becomes smaller, so the translocating structure needs to look a little bit more to find the empty spot, the oscillations, are a little bit more sparse. You can see down here, the same down here. And in fact, we had actually seen that. So if you look at a big structure being engulfed, this one here, you can actually see that the DFCP1 and the LC3, they don't all form in the same place, but actually you can see them forming in 3 different areas, and then they basically come together so that eventually you enclose the structure. So this kind of simulation predicted that the number of oscillations at the end of the process would be proportional to the diameter of the structure that's being targeted. And you can see this here. So at the end of the simulation, down here, 910 seconds, we have a nice linear relationship between the number of oscillations and the diameter of the structure. And in fact, this is exactly what we did when we actually measured the diameter of each of these mitochondria here and how many oscillations we observed. So we think that this sort of explains this behavior. And so just to summarize then, the difference between non-selective and selective autophagy, which in this case, I talked about mitophogy, it's very similar actually. So we still think that they provides a spatial clue that allows so this is the non-selective autophagy that attracts the machinery. We think a very similar thing happens in mitophagy, but in this case, the AR comes very early in order to sort of restrict the targeted mitochondrion. And in this configuration, we then see a hierarchical translocation of the different mitophagy and autophagy proteins, similar to what we see up here, but with the addition of the mitophagy adapters, and we see the same extrusion step where the forming autophagosome that has engulfed the mitochondrion now basically leaves the structure. So very similar. And the reason why we think you have this kind of pathway is because in this kind of mitophagy, you want to create a double membrane structure that's very tightly associating with the targeted mitochondrion. So this is an example from one of the EMs that Eva-Lisa made from our work. And this is basically 3 slices. And you can see how tightly the mitochondrion is being engulfed by the forming autophagosome. So we think that having this kind of restriction helps the machinery to engulf very tightly the structure. So if we take at one level the different components coming together in a hierarchical fashion, starting from ubiquitination, then the activation of TBK, the adapters, the autophagic machinery coming together, and then finally the rest of the machinery down here, we think that if we look at the forming structures that this machinery forms, there is a difference between engulfment by the ubiquitin or the association with the AR or the omecasome versus the adapters, for example, optinurin and the ULK complex exemplified here by ATG13. We think that these two are much more dynamic as they come on and off the structures. And the reason why we think they do that is because they guide where the rest of the machinery is going to go. And so you end up basically with a sort of immobilized structure that then has the translocated early machinery that then basically creates pieces of phagophore that get stitched together to generate the finally closed structure. And if you have this kind of pathway and this kind of machinery, then basically you can enclose all kinds of different structures, because the only thing you'd have to do is just make little pieces and then stitch them together until basically you have the whole thing engulfed. And so in our opinion, this is the biggest difference between non-selective and selective autophagy, this kind of sort of like modular response. And why is this the case? So we can divide the machinery for autophagy in basically 2 or 3 parts. So you have some machinery, the kinase, the protein kinase complex, the ULK complex, the lipid kinase complex, the VPS34, then you have the effectors of the lipid, which is the WIPI proteins. And so these are very simple translocations or kinase reactions that are very, very fast. But then you have a much slower machinery that needs to undergo many, many different reactions in order to end up with a LC3 that has been cleaved and then it has been lipidated. So a fast set of reactions here, a much slower machinery here. And it's basically like a fast car trying to drag, a much slower moving object. And if you make this too tight, it's not going to work, because I think this is going to want to go very fast. This has to go slow. So you can couple them, but you cannot make them too tight. And that's the idea behind this kind of machinery going on and off, marking the areas where the lipidation machinery will then come in order to generate the double membrane. And if you look very carefully at one of the EM images that I showed you at the very beginning, you can actually see that these 4 Salmonella particles, they are being engulfed here with a continuous phagophore. But look here, you can see a little phagophore that's coming together and being stitched. Following this, you can imagine that it's finding the adapters here, find this, and then another little phagophore here. So you can actually see that this is probably taking place not in our imagination, but in cells as well. And with this, I would like to stop. I want to thank you for listening to my talk, and I'll be very happy to answer questions.
That was great and very, very, very beautiful. So people are starting to write into the chat box. So maybe actually, Scott, if you can unmute, I don't know if you can unmute, you can ask your question directly.
Yeah, absolutely. That would be great.
Oh, okay. Thank you. This was fantastic. Hi, thank you so much for giving us a wonderful talk. I had two questions. First, this structural model that you've described with the omegosome and the ER contacts. Is this only related to ivermectin-induced mitophagy, or does it apply to the other forms of mitophagy that you described, such as if you induce mitophagy with a protonophore, or if you induce it with an uncoupler? And then my second question was related to: is there any relationship or requirement or interventions affecting ER mitochondrial tethering that might affect the formation of these membrane structures around the ER?
So the second question, I think definitely yes, but we don't have enough good data yet to show, but definitely the contact sites we think are very important. For your first question, so we have only looked at the CCCP and oligomycin and antimycin induced mitophagy as described by Richard Yule, but without overexpressing parking, because in my opinion and in our hands, parking overexpression makes a very difficult to interpret situation and model. So if you go to cells that have low but detectable parking expression, for example, HEX cells or MEFs have enough parking that if you put CCCP or oligomycin antimycin, you can see a little bit of mitophagy, not the kind that engulfs all of the mitochondria overnight, but just a little bit. So in these conditions, we see a similar kind of dynamic, both in terms of the ER providing the cradle, but also the oscillation, the oscillatory dynamics. But I must confess that we have spent most of the work using ivermectin. So we haven't done as much a detailed analysis, but the basic parameters, I think, are similar. And we haven't looked at any other type of mitophagy. The reason why we picked ivermectin is because, and I was reluctant for many years actually to go into this area, because I was worried that if we do something like CCCP that takes 6 or 7 hours to get going, by this time I was worried that the cells may be in such a state that you may be looking at a lot of, not very healthy responses. Ivermectin works within half an hour. And the cells are still okay while we are doing these experiments. So that's why we used it.
Nick, this is Peter. Can I ask you a question about the ATG2? Is the thinking that when ATG2 is connected, that there is a burst of new lipid synthesis at the ER, or just a flowing of pre-existing lipids that are going through that transport mechanism to enter the phagophore?
So I think that this is not completely settled, Peter, but I think the early data suggests that you may actually need new lipid synthesis during the induction of autophagy. And I think the idea may be that this may be used either for the lipidation reaction or for the transferring. But I don't know, Dan may want to address this point as well. But this is my understanding from some data that new lipid synthesis is important.
Let's let Dan talk, because he also has a question for you.
Well, let me ask this two questions, Nick. One was that your EM images were quite striking in terms of having rough ER in close opposition to the forming
Yes.
either the omegasome or the phagophore. Do you always see that?
Yeah, I would say 95% of the time.
And I mean, I don't recall seeing that in yeast, which is what I'm more used to looking to. Do you know if that similar mechanism is occurring?
Well, I think in yeast you have the two connections to the ER and the phagophore basically forming in between. So I think it's slightly different, but you still have the connection to the ER and maybe via the exit sites for yeast.
So let me go back to the mammalian for a second. So with that circular omegasome, you have the red interior.
Yes.
Is that essentially a sheet in non-selective autophagy? Is that a sheet of membrane initially across that circular opening?
I think so, because if you look very carefully at the movies, you can actually see that although, we depict it as a ring, but actually there is a little bit of structure and a little bit of signal inside. So
right.
Your next question is going to be, how do you maintain the, right? So this is something that the structural biologists like Roger Williams, they really hate this because it's very difficult to imagine how this happens unless the middle part where you see the lipidation reaction is so busy with a lot of protein complexes that basically the lipid cannot diffuse inside and it has to stay outside. And I don't know if you remember, there was a paper a few years ago saying that you have this mesh of proteins that basically are forming where the lipidation machinery is working. And that was based on some in vitro data. So that's sort of like my way out of it, but we don't really know. And it is kind of a weak point in the model. How do you, because we are saying that it's the same membrane, but it's just two different areas. Yes.
Nick, can I ask you this, Peter? Can I ask you the DFCP1 marker that you use? Is that selective, you believe, for this omegasome, or you actually see other puncta for other PI3P related processes that have nothing? Yeah, go ahead.
So we can do exactly the same experiment using WIPI2. So DFCP1 is not an essential protein in this pathway, and I'm embarrassed to say we still don't know what it is doing. But WIPI2, which is another effector of the pathway, is essential, and it's a PI3P effector, and it works the same place. So there's perfect colocalization between the two, and there is a sort of hierarchical relationship between DFCP1 and WIPI. If you knock out one, you accumulate the other structures. So it's not just DFCP1, but it's just because we had made a nice, stable cell line and people borrowed it for a lot of the experiments. It has been used by a lot of people, but it's not just DFCP1.
Okay, thank you. Vikram, is there a question? So I should just say that Nick also typed his own email address into the chat box. So if you have questions that you want to ask outside of the talk, you can email directly, but please go ahead and ask your question.
I have a question. Hi, I'm Mauricio. I'm a postdoc. I had two questions. One is related with the size of the mitochondria to be delivered to mitophagy. So my question is in some way connected mitochondrial fission with mitophagy, because it looked like both depend on the ER in this case. And And the other question is, in the case the induction, the mitophagy induced for FCCP, so is any requirement for ATP energy that in this case is not available to observe a massive mitophagy for this way?
So fission is absolutely required because we can block everything if we block fission. So that's very important for the first step. How physiological this is, I mean, you know, we by the time that we start imaging this process, we don't really have an intact mitochondrial network anymore. It's all pieces. But if we inhibit that, then we don't get the response. In terms of the ATP, I don't know. I don't know the answer to that.
Thank you.
Other questions?
Yeah, I'd like to ask one more, if I can. Nick, when you're talking about mitophagy, you have this large organelle that the ER can interact with, and going back to Scott's question, maybe involving tethering, etc. What about something like ribophagy?
We haven't looked. We haven't looked.
Can you speculate on how? Do you think it's going to be the same mechanism? Meaning you have to have a sort of a capture by the ER occurring first?
So, I mean, there is thinking that everything can happen on the surface of the targeted structure. So if you translocate the machinery there, you can basically, get it to work on that side. The problem with that is that, first of all, you're going to need to make membrane. So at some point, you're going to have to connect it to some membrane generating system. And the easiest way is to do it with something that is already known to be able to do that. And that is the AR. The other thing is that some of the components of the early steps, we know that they associate with AR. And so if they do that, they can work very nicely. So if you have the AR connection, you can rationalize how all of these components, if they find the adapters, they can basically do very similar, dynamics and reactions to what they do in non-selective autophagy. But this is just speculation on my part. We haven't actually looked at any other kind of selective autophagy.
Let me just get in one quick additional question here, if I can. Okay, so certainly in yeast and also in mammalian cells, there are data that implicate the Golgi or recycling endosomes and possibly the plasma membrane as sources of membrane. Yes. And in general, I think the view, certainly mine, is that in part there's vesicular addition of membrane, not all from this direct ER connection. What's your view about this, explaining those?
So I think, I'm a very big fan of Eva Lisa's work. And so I think it was published in Autophagy actually, where she sees connections with almost all, intracellular organelles to the phagophore. So absolutely, I think it's very likely that there is some contribution from a lot of different organelles. And, even if you look at our tomograms, you always see, a piece of mitochondria nearby. But I think the ER has a primacy in this. So I think the ER is always there, and then the rest, but it's probably not the only one ever. There's probably always some other contribution, which can be either vesicular, but they can also be contact sites. So I think, I wrote a review last year, and I said maybe the phagophore is just, the contact site where basically all of the membranes, make some connection. So I'm not dogmatic really at all about that.
So this is Peter. I'll take the liberty of asking you one more because, as you know, I originally asked you to talk a little about ERphagy, but if the ER is the source of the membrane that will be used for the autophagosome, and the ER is also the target membrane, then I guess you're envisioning that the ER bends back on itself, and that additionally, the horizontally in the plane of the membrane, there's segregation in a way that can create the target as well as create the recognition of the target.
Yeah, absolutely. And in fact, I showed an example of, so I showed the selective autophagy, where basically the idea is to make a very tight engulfment of the membrane. But I showed you an example where there was a piece of mitochondria and a piece of the ER inside the autophagosome. And this was non-selective autophagy, where, basically whatever is nearby, some of it is going to go in. So I see no topological problem to have the ER as the source, but also, to be part of it, to be engulfed. It won't be the same piece of the ER, but it can be, a nearby piece.
Right. Your internalized ER in that picture was ribosome-bound ER.
Yes.
The membrane that's being used to engulf, obviously, is ribosome-free membrane.
Well, maybe, but I think you can see ribosomes inside the, in non-selective autophagy, basically, I think anything goes.
Yeah.
As long as it's not too much, I think.
Yeah.
So you just need to sample a little bit of everything.
Any final questions before we thank Nick? Again, his email address is in the chat.
Link has a question.
Yeah, is it okay if I ask a question?
Please.
So, Nick, this is really wonderful talk, and I'm amazed by those images. And so my question to you is for those ERs, I'm really struck by the fact that our ER could wrap around the mitochondria and mediating this process, mitophagy process. Have you guys done anything in vivo, for example, the animal tissues on the fasting feeding conditions? For example, the liver?
No, we haven't done anything. We haven't done it.
On the regular feeding condition, we've done quite a bit of a TEM on the hepatocyte, on the liver cells.
Yes.
I don't ever remember seeing that. Certainly that we may not be looking at the right conditions, but I never seen a mitochondria wrap around by a piece of RER. Mm-hmm. I do remember seeing some very small mitochondria maybe wrapped around by a smooth ER, but not the rough ER. That would be really striking.
So this is spontaneous mitophagy you're talking about, without.
Yeah, without this starvation, for example, for a long time, just regular feeding condition. I guess.
Yeah, there is a kind of mitophagy that I didn't talk about, where basically I think you pinch a tiny bit of ER, and it could be a very physiological as well, where you don't want to make the mitochondria into fragments, and you don't want to get rid of all of the mitochondria, but you try to get rid of where the damage is. So that's a different kind of mitophagy, where you see very, very small pieces of mitochondria that are being engulfed. And we have actually imaged that also, and it's a very, and there's other people who have published on that. And in that case, it almost looks like you preform a phagophore that then, as a phagophore, it goes and attacks the mitochondrion, and then as it attacks, it basically closes. And I think it's a very different, maybe this is what you have seen.
So what is do you know the nature of the ER resident protein that's involved in your process, in the mitophagy process, that is required, for example, that like tethering?
So I think there isn't a single protein that does all of this. The idea from non-selective autophagy is you have a group of maybe 4 or 5 different proteins. For example, VAP-A, VAP-B, a little bit the SERCAS, and a little bit syntaxin 17, the PI synthase. So these are all ER-associated proteins that are part of the mechanism, but none of them is on its own responsible for it. And I would guess, based on the fact that we think the pathway ultimately is very similar, that the same kind of collection of proteins, none of them on its own absolutely required, but all of them in some ways required. Yeah.
Thank you.
Any last questions before we close?
I had another question, if it's possible. Mauricio again. My question is related with this no-selected autophagy. I was thinking in the concept of no-selected mitophagy, because, for example, we study brown adipocytes. When we put that mice in 30 degree, the mitochondria, healthy mitochondria, start to reduce the number. So my question is, how the cell decide what mitochondria to eliminate when is all the mitochondria are healthy? It's just by size, or there are another criteria? What do you think?
So it's a similar question that we have when we have a cell full of maybe 500 or 600 mitochondrial pieces, but only 3 or 4 are the targets at any one time. And I think that it has to be some kind of damage, maybe a small kind of damage that maybe induces a ubiquitination. So although you don't do anything to the mitochondria, but maybe there is some stochastic damage, and then the machinery is basically designed to sort of like zero in on this damage. That's what I think in the case of our pathway, and maybe I would think maybe potentially what you are seeing as well.
Okay, so in that context, the cell is not going to eliminate any healthy mitochondria.
Yeah, I mean, I think it will be easier to imagine that in the normal process of metabolism and so on, you're going to have some mitochondria that become mildly damaged, and that may be enough of a signal to get rid of those, or get rid of a piece of them, even not the whole mitochondrion.
Thank you.
Well, thank you so much, Nick. This week you're a cheap date because you're coming all the way from England, beamed by cyberspace, and then you get to eat your own dinner on your own. But we're really glad you had a chance.
It was very enjoyable. Thank you for the invitation, and good luck to the
I
election.
hope we see you again. Yeah.
Thank you, Michigan. All right. Take care. Bye.
Take care. Bye-bye.
Bye, Dan.